Hello everyone, my name is Anindita Sekia, a student of 3rd Professional Year BVSC and AH of Lakhimpur College of Veterinary Science, Joyhing, North Lakhimpur, Assam. And in this video, I am going to describe the neurohumoral transmission and synthesis, storage and release of neurotransmitters in adrenergic and cholinergic transmission. The contents of the video are nervous system, neuron, neurohumoral transmission, components of neurohumoral transmission, events of neurohumoral transmission, synthesis, release and storage of transmitters in cholinergic division, and synthesis, release and storage of transmitters in adrenergic division. The first topic we have is the nervous system. It is the system that controls the functioning of all other systems in the body. It is divided into the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord, and the peripheral nervous system is further divided into the somatic nervous system and the autonomic nervous system. The somatic nervous system is in control of all the voluntary actions of the body, and the autonomic nervous system is in control of all the involuntary actions of the body. It is further divided into sympathetic division and parasympathetic division. The sympathetic division mainly is concerned with the fight and flight reactions, and the parasympathetic division is concerned with rest, digestion, etc. Next, we have the neuron. The neuron is the basic functional unit of the nervous system. It transmits information to other nerve cells, muscles, and glands. It consists of a cell body, axon, and dendrite. The cell body consists of nucleus and cytoplasm. Dendrites, these are extensions from the cell body and receives electrical signals. There is another extension of the cell body which is known as the axon, which ends into axon terminals. The axon terminals consist of synaptic vesicles which stores the neurotransmitters, which actually helps in transmission of the impulses from one neuron to the other neuron, or from one neuron to the effector organ, gland or muscle. Then we have the neurohumoral transmission. It is the transmission of impulses through synapse and neuroeffector junction by the release of humoral chemical substances. These humoral chemical substances are the neurotransmitters, which will be dealt on in the later slides. Then the components of the neurohumoral transmission are pre- and post-ganglionic fibers, ganglia, neurotransmitters, neuroeffector junctions or synapses. So the first components of the neurohumoral transmission are the preganglionic nerve fibers. These are the fibers from the CNS to the ganglion. They are cholinergic, that is, they use acetylcholine as the neurotransmitter, and they are always myelinated. Then we have the postganglionic fibers. These are the fibers from the ganglia to the effector organ. The neurotransmitters they use are, in the parasympathetic division they use acetylcholine, and in the sympathetic division they use noradrenaline. Then the next component is the ganglia. It is a group of nerve cells in the peripheral nervous system. It consists of nerve endings of the preganglionic fibers and cell bodies of the ganglionic fibers. Then we have the neurotransmitters. These are the chemical messengers which helps in the transmission of impulses between neurons or between neurons and the effector organs. These are present in the synaptic vesicles of the nerve terminals, and when an action potential arises at the nerve terminals, these synaptic vesicles fuse with the axonal membrane, and these neurotransmitters are released in the synaptic cleft. These neurotransmitters can either be excitatory or inhibitory. Here is a list of some excitatory and inhibitory neurotransmitters. The excitatory ones are glutamate, aspartate, noradrenaline, adrenaline, histamine, and acetylcholine. And the inhibitory ones are GABA, glycine, and serotonin. Then the neuroeffector junction. It is the site of transmission of impulses between two neurons or between a neuron and a gland, organ, or muscle. So now we are going to learn about the steps involved in neurohumoral transmission. It includes impulse conduction, transmitter release, transmitter action on post-junctional membrane, post-junctional activity, and termination of transmitter action. The first step is impulse conduction. So, before the arrival of a stimulus, the cell is said to be in its resting state, and at this stage, the concentration of sodium ions is higher in the outside of the cell, and there is a higher concentration of potassium ions inside the cell, and the inside of the cell is comparatively more negative than the outside of the cell, and there is a voltage difference of minus 70 millivolt between the inside and the outside of the cell, and this voltage difference of minus 70 millivolt is said to be the resting membrane potential. And as soon as there is an arrival of an electrical impulse to a nerve fiber, it causes a sudden increase in the sodium permeability, and as such, sodium ion enters the nerve fiber through sodium channels and changes the polarity of the inside of the cell, that is, depolarizes the cell. And as soon as there is depolarization, the potassium ions that are present inside the cell starts moving out of the cell to the outside, thus restoring the polarity of the cell, and this is called as the repolarization. This cycle of depolarization and repolarization is what we call an action potential. And as such, through the process of depolarization and repolarization, a nerve impulse travels through a nerve fiber. And now we'll see how some drugs affects impulse conduction. Drugs such as tetrodotoxin and saxitoxin decreases sodium permeability, blocks axonal conductivity and causes flaccid paralysis. Drugs such as batrachotoxin and scorpion toxin increases sodium permeability, causing persistent depolarization resulting in spastic paralysis. Local anesthetic agents tend to block axonal conduction. The next step in neurohumoral transmission is transmitter release. The neurotransmitters are stored in the prejunctional nerve ending within the synaptic vesicles. As soon as a nerve impulse reaches the nerve endings, it causes the fusion of vesicular membrane and axonal membrane through calcium entry, and all the contents of the synaptic vesicles are released into the synaptic cleft through exocytosis. This release of neurotransmitters can be modulated. It can be modulated either by itself or by activation of a specific receptor interaction on prejunctional membrane. Example, norepinephrine release is inhibited by norepinephrine via alpha-2 receptor, dopamine, adenosine, and prostaglandins. Acetylcholine and isoprenaline increases norepinephrine release. Then the next step is transmitter action on post-junctional membrane. The released neurotransmitters and binds with the specific receptors present on the post-junctional membrane, and depending on its nature produces an EPSP or IPSP. So what is an EPSP? EPSP stands for excitatory postsynaptic potential. So what happens is, whenever an excitatory neurotransmitter is involved, it causes either propagation of impulse in the postganglionic neuron or stimulation of an effector organ. And now, what is an IPSP? An IPSP stands for inhibitory postsynaptic potential. So whenever an inhibitory neurotransmitter is involved, it binds with the specific receptors and increases the permeability to potassium ions and chloride ions, and this causes hyperpolarization of the cell. This stabilizes the postsynaptic membrane, increasing the threshold to stimuli, and elicits an inhibitory response. Now the last step in neurohumoral transmission is termination of transmitter action. After combination of the transmitters with the specific receptors in the postganglionic membrane, they have to be terminated. In case of acetylcholine, it is rapidly hydrolyzed by enzymes called Cholinesterases. There are two types of cholinesterases that hydrolyzes acetylcholine into acetate and choline. These cholinesterases are acetylcholinesterases and butyryl cholinesterases. In the case of norepinephrine, it is taken back into the preganglionic neuron by membrane-bound norepinephrine transporter and recycled for further use. So now we are moving on to the next part of the presentation. That is, now we are going to learn about the synthesis, storage and release of neurotransmitters in the cholinergic division. The major neurotransmitters in the cholinergic division is acetylcholine, and it is synthesized in the mitochondria of the cholinergic nerve endings. So what happens in the mitochondria is, there occurs a reaction between ATP, acetate and coenzyme A to form acetyl-CoA. This acetyl-CoA, after its formation in the mitochondria, travels into the exoplasm. And at the same time, choline enters into the exoplasm through the axonal membrane. And there occurs another reaction between acetyl-CoA and choline in the presence of the enzyme choline acetyltransferase to form acetylcholine. There are certain factors that affect the transfer of choline into the exoplasm, and these are: it is directly proportional to the extracellular sodium concentration and it is inhibited by hemicholine. Now, storage of acetylcholine. Acetylcholine is stored in the synaptic vesicles, and also some free acetylcholine is found in the cytoplasm of cholinergic nerve terminals. Release. So what happens is, whenever an action potential arrives at the cholinergic nerve terminals, it causes the depolarization of the area. This opens up calcium channels, which leads to the fusion of the synaptic vesicular membrane and the axonal membrane. And as such, acetylcholine is released into the synaptic cleft through exocytosis. There are certain inhibitors of the release of acetylcholine, which are: an excess of magnesium ions, botulinum toxin or procaine causes inhibition of the release. Also, black widow spider venom, it first causes a release of excessive amounts of acetylcholine, which is then followed by blockade of release. Now, the fate of acetylcholine. After this neurotransmitter combines with the specific receptors on the postsynaptic membrane, it has to be terminated. So, what happens is, acetylcholine gets hydrolyzed into acetyl and choline with the help of an enzyme cholinesterase. There are two types of cholinesterases. These are acetylcholinesterases and butyryl cholinesterases. And now we are going to learn about the different cholinergic receptors. There are two types of cholinergic receptors, which are nicotinic and muscarinic, which are again divided into various subtypes. The subtypes of nicotinic receptors are NM on skeletal muscle and NN on neuronal cells. The characters of the nicotinic receptors are given in the slide. The next type of cholinergic receptor are muscarinic receptors. There are actually 5 subtypes of muscarinic receptors: M1, M2, M3, M4 and M5. But M1, M2 and M3 are the major receptors, and the characters and functioning of the receptors are given in the slide. So now we move on to the last part of the presentation, where we are going to learn about the synthesis, storage and release of neurotransmitters in the adrenergic division. Noradrenaline or norepinephrine is the main neurotransmitter in sympathetic or adrenergic division. Others are dopamine in the brain and epinephrine, which is secreted by adrenal medulla. These are collectively called as catecholamines. Synthesis of catecholamines. Catecholamines are synthesized from the amino acid phenylalanine. In the pathway, we can see the conversion of tyrosine to dopa is a rate limiting step, and the enzyme tyrosine hydroxylase, which catalyzes this reaction, is the rate limiting enzyme. And its inhibition by alpha methyl paratyrosine leads to depletion of catecholamines. Noradrenaline is synthesized in all adrenergic neurons, and adrenaline is synthesized in the adrenal medullary cells. Storage of catecholamines. Both noradrenaline and adrenaline are stored in the synaptic vesicles of adrenergic nerve endings. Release of catecholamines. Release of catecholamines is coupled with a nerve impulse, and this release is autoinhibitory in regulation. And now we move on to the termination of the catecholamines. Unlike cholinergic junctions, there is no synaptically located enzyme that degrades the catecholamines. Instead, reuptake is the main mechanism of the termination or inactivation. The reuptake takes place as follows. Uptake 1. 60% of norepinephrine re-enters into the synaptic vesicles of nerve endings. Uptake 2. 20% is diffused into extraneuronal sites. 5% binds with the receptors and about 15% is destroyed by monoamine oxidase. In the liver, it is metabolized by catechol O-methyl transferase. Now, the adrenergic receptors. Adrenergic receptors are membrane-bound G-protein coupled receptors which function primarily by increasing or decreasing the intracellular production of second messengers CAMP or IP3 or DAG. There are two main types of adrenergic receptors, alpha and beta. Two alpha receptor subtypes, alpha 1 and alpha 2 are present, and three subtypes of beta receptors, beta 1, beta 2 and beta 3 are present. So, this is the end of the presentation. I hope you liked it and thank you for watching.